The last of the topics we're going to be covering with respect to the immune system is diseases that are a result of either the immune system itself not functioning correctly because of a genetic problem or the immune system not functioning correctly because of some other problem. And these are called immune deficiency diseases. So there are two main types of immune deficiency diseases or immunodeficiency diseases, and they're roughly categorized as being either primary or secondary. Primary diseases are also sometimes called congenital diseases, and that's because congenital literally means with birth. So these are diseases that occur at the time of delivery because of some underlying genetic condition that has been present since the fusion of egg and sperm. And we'll talk about a couple of examples of these. The other class of immunodeficiency diseases are secondary diseases, and these are not do not have a genetic underpinning, but rather they result from some other type of factor. And we'll talk about one example of that. So let's first start out looking at primary, and I'll give you a few examples of genetic disorders that impact immune system function. Now, if you remember, I want to just have you look here at this flow chart. This is showing you, just to remind you again, the development of B and T cells that occurs in bone marrow. And we start out with a pluripotent stem cell, and it gives rise to a lymphoid precursor cell that can give rise to either what will become B cells if we're over on this side, or what will eventually become T cells if we're over on this side. Now, primary immune deficiencies oftentimes result simply from one genetic defect, just one gene having a defect in it. If it's in the wrong place, can inhibit proper development along one of these pathways, and it can be highly have a huge impact on the body, and in fact, a lot of times it can be deadly. Now, there are different types of primary immune deficiencies. Sometimes the type of B cells impacted by the genetic defect are the B cells. Sometimes it's the T cells. And I think you can see from this development chart, you can understand why maybe B cells might be impacted versus T cells based on whether it was a gene that had a defect in the development of B cells here, or whether it was a gene that had a defect in the development of T cells here. Some people, unfortunately, can develop a condition where they are unable to produce either B or T cells functionally. Some people, their B and T cells are pretty okay, but they have a problem with complement production. And some people have a phagocyte problem due to a genetic defect. Now, I'm only really going to focus on the B and the T cell deficiencies here, just to give you a couple of examples. So an example of a B cell deficiency, an extreme example, I guess would be the best way to say it, is X-linked A-gamma globulin anemia. And it's hard to say that. I want to remind you that antibodies can be referred to by a number of different names. Antibodies are also called immunoglobulins. You guys know that already. But they can also be called gamma globulins if they are free-floating in the blood. So X-linked A-gamma globulin anemia means that we have a defect found on the X chromosome, one of the genes on the X chromosome, and it's causing no production, that's what the A means, no production of gamma globulin in the blood. So this tells you that we're looking at a problem of B cells. This is also referred to in the old textbooks as Bruton disease. So if we take a close-up look at that same flow chart I showed you on the last slide, you can see here that there's a portion of the development here where we have what's called a pro-B cell that develops into a pre-B cell. And the big event that's happening here is the generation of heavy chains. Remember that immunoglobulins, antibodies, have a heavy chain and they have a light chain to them. Well, during the development of a B cell, the first genes that get activated are those that are responsible for the manufacture of the heavy chain, and then secondly, genes that are responsible for the manufacture of the light chain are activated. And in this kind of condition, in Bruton disease, the heavy chains get manufactured all right, but the light chain is not manufactured correctly. So we can't progress to the next step here. There's no way to. And as a result, I end up with all these pre-B cells. These are not functional B cells. They do not have the ability to manufacture functional antibodies. So obviously, you would expect a person with this disorder to have very few or no functional B cells. They're not going to have plasma cells because if plasma cells are derived from B cells. But on the other hand, their T cell response should be relatively normal. And you often see this disease in males because it's X-linked. Remember, males only get one X chromosome, so if they get a defective copy of the gene responsible for the manufacture of that light chain, they're going to develop Breton disease. And the other thing to notice about this is that because the T cell response is pretty well normal, any kinds of infections that normally are the responsibility of the T cell mediated response are going to get cleared fine. So things like viral infections typically will be cleared okay, fungal infections, protozoan infections. Those are really the domain of T cells, and they do a fine job of clearing those out. But bacterial infections become a real problem for babies born with this disorder. It usually appears around 6 months of age after maternal antibodies have completely broken down. Remember, we got IgGs that could pass from placenta to fetus. Once those are all depleted, then they start getting recurrent and chronic infections, things like otitis media, which is middle ear infection. Very painful for a little kid. Sinusitis, bronchitis, pneumonia, pharyngitis. All of those are inflammations or infections associated, you'll notice, with the respiratory tract. The respiratory tract is a big mucosal area. And remember that B cells are often found in mucosal areas, so it should make sense that infections that preferentially set up shop in mucosal areas would be the ones that would predominate in an individual with this disorder. Another example that's not as extreme, but does still involve B cells, is something called common variable immunodeficiency. And I kind of think of this as sort of a catch-all category because there's a bunch of different variations that fall into this category. Basically what it is a not so severe version of Breton in a way. You can think of it like that. It's highly variable for some individuals. They have a very moderate case of it where they fight off most infections pretty well. Other individuals can have very severe forms that really impact their ability to fight off infections. I'm not going to say too much more about that one. Another type of B cell immune disease is selective immunoglobulin A deficiency, and that should tell you exactly what it is. This is a case where we have B cells that can make immunoglobulin M, they can make immunoglobulin G, they can make immunoglobulin E, etc. But their ability to make immunoglobulin A is compromised. They cannot make immunoglobulin A. And as a result, they tend to have more infections in locations in the body where immunoglobulin A would normally be found in high concentrations. Once again, mucosal membranes tend to be a big one. So there are lots of different symptoms that can occur from common variable. I'm not going to worry too much about the selective Ig deficiency symptoms, but in common variable immunodeficiency, some examples of symptoms, and again, depending on what type you can have different symptoms. Some people develop arthritis. Imagine a little kid with arthritis. Some get autoimmune diseases. And some even get cancer. Now, what about on the other end of the spectrum? What happens if we look back at that developmental chart and instead of going down the B cell side, we go down the T cell side? What happens if I have some sort of a genetic defect that inhibits my ability to properly manufacture one or more types of of T cells? Well, then we've got a reduced ability to fight off things like viral infections, fungal infections, protozoan infections. Now, before I talk about one example of that called DeGeorge syndrome, I want to just remind everybody that there are two different types of helper T cells, Th1 and Th2, also called CD4-1 and CD4-2. CD4-1s are the helper T cells that are mainly responsible for for interacting with the cytotoxic T cells in the cell-mediated response. CD4-2s are the principal helper T cells responsible for interacting in the antibody-mediated response, just so you know the difference there. Now, here's one example that I can give you of a T cell deficiency called the DiGeorge syndrome. So here's our precursor lymphoid cell. It gives rise to a pro-T cell. Everything's going along fine. We make an immature T cell, and at this point, normal development stops. And the reason for that is we have little or no thymic tissue present. So this is from a baby that's born with a partial lack of a thymus, or sometimes they aren't born with any thymus at all, in which case it's not a good prognosis. So they have a very low amount of or no T cells, and as a result, they're really prone to viral, fungal, and protozoan infections. Meanwhile, their B cells are functioning relatively okay, so they'll be likely to be able to fight off some bacterial infections, although because the role of CD4 cells is so important to being a confirmatory step to antibody-mediated immunity, they're not going to have the most robust B cell response either. Now, some individuals with this, particularly if they have partial thymus, they can over time grow a larger bit of thymus, and they can actually partially or even completely recover from DiGeorge syndrome. But if they're born without a thymus, it's like I said before, it's likely they may die of the disease at a young age. Sometimes people get a really bad genetic double whammy. They can have what are called combined B and T cell deficiencies. And you might imagine that if we go back here to this chart, we're talking about some sort of problem up in here, some sort of genetic defect that's preventing the generation of the common lymphoid precursor, or maybe some sort of defect that's preventing that precursor from giving rise to the pro-B and pro-T cells. And the example of this that's always given in textbooks is severe combined immunodeficiency syndrome, or SCID. Awful, awful disease. People that have this disease literally have to live in a plastic bubble where they are completely isolated from the external environment. Their parents cannot touch them. They have to touch them through little glove-like extensions that extend into the bubble. There's about 20 different subtypes of this, and depending on which subtype a child has, it might be that their T cells are impacted, their B cells are impacted, their natural killer cells are impacted, or some variation or combination of impact among those. So these guys, because they have both problems with B and T cell function, are highly, highly susceptible to candida infection. That's yeast infections. Candida albicans. And they're also really susceptible to Pseudomonas, which is a bacterium, and a lot of other types of bacteria. Infections are a real problem. Another example of a combined B and T cell deficiency is Wiskott-Aldrich syndrome, this guy down here. And this is another disease that's X-linked, it's recessive, and what happens in Wiskott-Aldrich is there's a lack of a certain, there's the gene that normally functions to manufacture an enzyme called adenosine deaminase is not functional. And without that present, the body is not able to degrade purines. Purines are one of the two types of nucleotides found in DNA and RNA. And if that adenosine deaminase is absent, then the purine metabolite toxins start to accumulate, and these are very deadly to developing B, T, and NK cells. So what happens is, because of some deficiency elsewhere, it's causing a buildup of metabolic waste products that kills B, T, and NK cells. This is another example of a condition where, if not treated, the kid dies very, very early in life. And even with treatment, the treatment is often not successful. Really, the only course of action to try is to try to do a bone marrow transplant to give them some functional cells that can create adenosine deaminase. And they've had some success with that, but they've also had some fallout issues with it. So it's actually in a holding pattern right now. That's enough on primary. Next we'll look at secondary.